Welcome viewers, our topic today is on understanding gene function. In this video, we will explain the role of genes, its regulation and applications arising from genetic manipulation. If you are new here, welcome. We are a group of retired professors sharing bite-sized videos in life sciences. Let's dive into the topic for today. Gene insights. Understanding gene function is a fundamental aspect of genetics and biology. It involves the study of how genes control the development, growth, and behavior of living organisms. The knowledge of gene function is crucial for understanding the underlying mechanisms of various diseases and for the development of new treatments and therapies. Understanding gene function. The study of gene function begins with identifying the specific genes that are involved in a particular process or disease. This can be done using a variety of techniques including genome-wide association studies, transcriptomics, and proteomics. Once a gene has been identified, researchers can then study its function by manipulating it in various ways, such as by knocking it out or overexpressing it. Gene knockout and knockdown. Gene knockout and knockdown are two powerful techniques used to study gene function in living organisms. These techniques involve the intentional disruption of the function of a specific gene in order to study the effects on the organism's development, growth, and behavior. Gene knockout. Gene knockout is a technique in which a specific gene is completely removed or inactivated in a living organism. This can be done by several methods such as homologous recombination, where a specific DNA sequence is replaced by another, or by the use of CRISPR-Cas9, a genome editing tool that can precisely cut out or alter a specific DNA sequence. Gene knockdown. Gene knockdown, on the other hand, is a technique in which the expression or activity of a specific gene is reduced but not completely eliminated. This can be done by several methods such as RNA interference, RNAi, where small RNA molecules bind to and silence specific genes, or by the use of chemically modified antisense oligonucleotides, ASOs, that can bind to and silence specific genes. Applications. Both gene knockout and knockdown techniques have been widely used in various organisms such as mice, fruit flies, and zebrafish to study the function of specific genes in different biological processes. These techniques have been used to study the genetic basis of various diseases such as cancer, heart disease, and neurological disorders. Taken together, gene knockout and knockdown techniques have also been used to produce genetically modified animals and plants with specific desirable traits, such as resistance to diseases or improved nutritional value. This has the potential to improve food production and reduce the use of chemical pesticides. Genetic manipulation. One of the most widely used methods for studying gene function is genetic manipulation, which involves making specific changes to the DNA sequence of a gene. For example, in a technique called CRISPR-Cas9, researchers can make precise changes to the DNA sequence of a gene, allowing them to study the effects of these changes on the function of the gene. Gene regulation. Another important aspect of understanding gene function is the study of gene regulation, which involves understanding how and when genes are turned on and off. This is controlled by a variety of factors including epigenetics, which involve chemical modifications to the DNA itself, and transcription factors, which bind to specific regions of DNA and control the expression of genes. Bioinformatics analyses. In addition to these basic research methods, there are also various computational and bioinformatics tools that are used to analyze and understand gene function. These include gene ontology, which is a database that categorizes genes based on their function, and pathway analysis, which is used to understand how different genes and proteins interact in a particular process. Closing perspectives. As a quick summary, understanding gene function is crucial for developing insights towards the underlying mechanisms of various diseases and for the development of new treatments and therapies. The study of gene function involves identifying specific genes, manipulating them, and studying the effects of these changes on the function of the gene. Moreover, understanding gene regulation and using computational and bioinformatics tools also play a vital role in understanding gene function. The knowledge of gene function is an ever-evolving field with new discoveries leading to more accurate diagnosis and treatments for various diseases.